In this video, I'm going to show you how to press and mount plant samples to start your very own herbarium. Plant samples are collected and then stored in an herbarium. What is an herbarium? Let's sit down with botanist Dr. Chrissy McAllister to learn more about how she uses them in her research.
Yeah, well, herbaria are incredibly important. They are basically big museums. They just don't take up that much space because all of the samples that are in the museum are squashed flat, right, and glued on pieces of paper. But some of the biggest herbaria in the world, they have millions and millions of specimens. I mean, we're talking like 8 to 12 million specimens in some of the major herbaria. And they span a time frame from, I mean, I think some of the oldest ones, the samples are 100 years old, approaching 200 years old in some cases, if you take care of them. Wow. So that represents an enormous record of where plants were growing in the world at particular periods of time. It's a time record too, which is really valuable. So there's a ton of information that's just kind of laying there in herbaria, and there are a lot of people interested in going in and finding what now. You can also if you get permission from your director, you can take little bits and pieces, do destructive sampling, and get DNA. So depending on the quality of it and how it's been preserved, you can get some DNA out of it, and that's been a whole new source of knowledge for people as well, and data. So I've been working on a project about grasses and grass biology. I have a collaborator who's really interested in the evolutionary history of grasses. We're looking at one particular group of grasses. It's a group that's about 1,200 species. And the reason we focus on those is because in those 1,200 species, there are quite a number of them that are ecologically important, like corn and sorghum and things like that, sugarcane. And then there's also a whole bunch that are ecologically important. So on almost every major continent, if you see big grassland ecosystems, when you look at the species of grass that dominate those systems, they're almost always in this particular group of grasses that we look at. So that raises a really interesting evolutionary, like, why did that particular group, out of the tens of thousands of species of grasses, why did that evolutionary branch of the grass family tree do so well? Domesticate a lot of them, that they take over ecosystems, right? So there's no silver bullet answer to that question, but there's a lot of little things that we can look at, including the anatomy of their seeds, the anatomy, the shape and structure of how they reproduce themselves. And how do you need to do that, you need really big sample sizes, and you need them from all over the world. So awesome if we had the time and money and resources to like fly people all over the world and collect that material, because that's cool field work, but we don't. So we send people to the herbarium. You can pull out samples, like I said, from across the world and across time. And if you're lucky, you can pull genetic material out of them as well.
What a great idea to, instead of going around the world, people have already done that. And it's a great resource for scientists to use for a variety of projects, including yours.
Exactly.
Great. Well, thank you.
Here are a few of the poisonous plants you'll want to avoid. The first is poison ivy, Toxicodendron radicans. Poison ivy has leaves of 3, with the terminal leaf on a leaf stalk. Slight margin serration with large teeth at the lower edge of the 2 leaflets. It can grow as a shrub or a vine, and the vine has hairs, as you see here. Poison oak, Toxicodendron diversilobum, a plant that is similar to poison ivy, but its leaves are shaped similar to oak leaves, hence its name. Leaflets are duller green than poison ivy and usually more distinctly lobed or toothed. Leaflets have hairs on both sides, unlike poison ivy. Poison sumac, Toxicodendron vernix, 7 to 13 leaflets in pairs with a lone leaf at the end. Leaves are oval, elongated, and smooth edge, usually 2 to 4 inches long. They are bright orange in the spring, dark green in the summer, and red-orange in the fall. Poison ivy, poison oak, and poison sumac all have a chemical known as urushiol. Urushiol is an oily substance composed of organic compounds that have allergens, which can cause an itchy rash. If you think you might have touched one of these plants, scrub the area hard with soap and warm water. Think of the oil like grease from your bike chain. It is not easy to remove. Giant hogweed, Heracleum mantigazeanum, has huge leaves, incised, deeply lobed, up to 5 feet across. It has white flowers with 50 to 150 flower rays clustered into an umbrella-shaped flower. The stem is green with purple splotches and coarse white hairs. Thick circle of hairs at the base of the leaf. Flowers in late June to mid-July and can grow between 7 and 14 feet tall. You do not want to touch this plant. Stinging nettle, Urtica dioica, has soft green leaves that are 1 to 6 inches long, oppositely arranged. The leaves are strongly serrated along the margin and have a terminal tooth that is much longer than the adjacent lateral. Now I want you to pause this video. Search online for the term common poisonous plants, plus your area, for example, Illinois or California or Washington, wherever. And I want you to draw these leaves of the most common poisonous plants in your nature journal under a heading called Poisonous Plants. Please draw at least 6. Avoid toxic garden plants that you might find and really focus on natural plants that are in ecosystems that you are exploring. It is fine to repeat some of the same species I just shared with you if that would be helpful. Pause now and I'll wait. After you understand which plants you want to avoid because they're poisonous to you, you want to look at which plants are rare, threatened, or endangered in your area and avoid those as well. Go to Google and type in rare, threatened, and endangered species of your state. Usually you'll find a Fish and Wildlife Service website that you can click on, and you can look at federally threatened endangered species for your state. It also tells you the range of those species, including counties. So find plants, but it's good to look at all these other ones too. And I want you to draw 3 or 4 of these plants just to become aware of them. If you click on one of these species, it will take you to a website. You can click on the Plant Home website, and often you'll find pictures of the species. Use these pictures to base your drawings from. Now pause this video and search online for rare, threatened, and endangered species, plus your area. Draw the leaves of these plants in your nature journal under a heading called Protected Plants. Draw and label 4 of these plants, and then go to a website associated with your state's Department of Natural Resources, DNR, and peruse some of the state protected plants. Pause right now and I'll wait for you to do your drawings. Wherever you are, make sure you have permission to collect plant samples. Some areas, like national parks and nature preserves, do not allow the collection of plants or anything else for that matter. So make sure you have permission, especially if you're on private property. When you're out collecting plant samples, there's a few materials you'll want to take with you. The first thing is your nature notebook, so you can make recordings. Second is a pen. You also want to bring masking tape so you can label all of your specimens, and then simply a bag to carry them in. In addition, you might want to bring along some shears or some sort of cutting implement so you can make a nice clean cut of your sample. And then of course you'll want your smartphone that has a camera and a GPS built into it. If you're on a long hike and you don't want your plant samples to get damaged on your way back, you can put them in your nature notebook. Here I am in the woods collecting my samples. So I'm going to look for a representative sample from this tree. A leaf that isn't unusual. Not too big, not too small, not weirdly shaped. something that looks like all the other leaves. Also, when I take a sample, I want more than just one leaf, especially if they're small. I'll get as many leaves as I can get on a page. That said, if it is too big, maybe you can only do one leaf, and you might even have to fold that leaf to get it onto the page. Also, you want to grab some of the twig that the branch is on. And the reason you want to do this is so that you can identify the branching pattern, whether it's opposite or alternate or whorled or whatever it might be. So these leaves look fairly representative, and so I'm going to make my cut right here. And that's more than I need, certainly, but that's okay, because I will pull a few off, and when I press them, I will select the best and most representative leaves to use. Some of them have bite marks in them, and so I probably won't use those. I'll just use the ones that look the most representative. So now that I have my Quercus alba, that's white oak by the way, I'm gonna label it so I don't get confused as to what it is. So I just took some masking tape, and here I'm just putting on a label, and I'm gonna call this one number 1. Number 1. All right, so there it is, number 1. And now I'm gonna put this in my plant collection bag. All right. Like I said, if I was on a long hike, I might try to press it just to preserve it, but I'm pretty close to home, so I'll just use a bag for now. Okay, so now that I have number 1, I'm gonna take some data about the location in which I found this plant sample. So I'm gonna grab my nature notebook. So I have my nature notebook here, and I'm gonna turn to my plant collecting page. All right, and I'm gonna start filling it in. So the first category is ID. Remember, this was number 1. So I'm gonna write number 1. Common name, white oak. Scientific name, Quercus alba. Location, well, I am up in Michigan, lucky me. So this is in Glen Arbor, Michigan. So just the general location. And we are in the Leelanau County. All right, now I need the latitude and longitude. And for latitude and longitude, that is pretty easy to find on your smartphone. So I'm gonna go into my smartphone. There's a folder called Tools, and there's a compass. So I click compass, and at the bottom of my compass is the longitude and latitude of my location. So I'm gonna write those numbers down in my nature notebook. So when I'm finished, I have a sample that corresponds to this entry in my nature notebook. This information that's in my nature notebook is gonna go on the herbarium sample once I mount the plant after I've pressed it. You know, one of the interesting things to look at when you're out collecting samples is the location the tree is growing. You can look at the topography. What kind of slope is it on? Is it flat like we are right now? Or is it on a steep slope? Is it on a ridge top or a valley bottom? Is it on a north facing slope versus a south facing slope? What is the soil like? Is it clay soil? Is it sandy soil? Is it rocky soil? Those are all really great pieces of information to capture when you are looking at a tree and its environment. So that is the physical environment, and then there's the ecological component. And so one thing you can look at is, who is it growing next to? I see some white pines, I see some black cherries, I see some other white oaks, and so that gives me a sense of the community in which this tree is growing. Make these observations, and pretty soon you will start to see patterns emerge. Now that I've collected my samples, it's time to press and dry them. And for that, we only need 2 things: old newspaper and cardboard. We're gonna make a plant sandwich. And in the middle, the peanut butter and jelly is going to be the plant itself. So here I have a stack of cardboard and a bunch of newspaper that has these half sheets. So what I'm going to do is I'm going to take one of my labeled specimens. Here's my white oak. And I don't need all of this. And so I'm going to break some of it off so that it presses neatly. And what I am trying to achieve here is not too much overlapping. So I want it to dry in the way I press it. So I'm spreading them out. I don't want too much overlapping. And I will fold the newspaper over. And so now there's 2 pieces of newspaper, one on each side of the sample. And then I place it in a cardboard sandwich. So I have a piece of cardboard and then another piece of cardboard over the top of it. So I have cardboard, newspaper, plant sample, newspaper, cardboard. So now that we have our cardboard sandwich, we want to apply some weight to this and let it dry for at least a week. You can use rocks, furniture, heavy books, but you want to make sure that it is flat and evenly pressed. If you do not let your plants dry for a full week, there is a chance that when you go to mount them, they will continue to dry and shrivel and they look a complete mess. So letting your plants dry fully out is fairly important. Once your plant samples have dried, it's time to mount them. So it's been a week now and we're ready to mount our pressed and dried sample. For this, you'll need your sample. In addition to paper, I only have normal printing paper, and so I'm going to use masking tape to tape my 2 pages together. A paintbrush, glue, a container to mix the glue with a little bit of water to water it down, and a pen to take all the information in my nature journal and copy it onto the paper of the sample. So I'm going to start by making my glue recipe. So I'm just taking normal white glue and I am just going to water it down slightly. And there's no exact recipe here. You just want it to be a little bit more runny, not as concentrated. So now I'm going to tape my 2 pieces of paper together to make an 11 by 17 sheet. So there we go, I'm gonna flip it over. And now I'm ready to glue my leaf arrangement onto the paper. And one pro tip I have for you is to flip one leaf upside down so that you can see the back side of the leaf. And so this is how I'm going to mount it, is like that. So I'm going to flip this over and I'm going to apply glue all around the leaf. And again, it's very important to make sure that your leaf is dry. You want to get it all the way to the edges. And once we're done with this, we're going to repress it between the newspaper so that it evenly attaches to the paper. All right, and then this is the leaf we're going to do upside down. So getting glue everywhere. And this leaf we're not going to use. I'm going to take this leaf off. All right, so now I'm ready to mount. I bring it over. And I don't want any of the leaves to be going off the page. And I don't want them to be folded or overlapping. So just like that. I can write down all of the unique identification information right here. So this was sample number 1, Quercus alba, white oak. Once I'm finished gluing and documenting my sample, I put it back in here and I let it dry overnight, and it'll be ready to go by tomorrow. You can put a little weight on it, like your nature notebook, and hold it down steady. As a result of watching this video, you should be able to understand the use of herbaria in research. You should know how to collect and label a plant sample. You should be able to press and dry a plant sample so that it can be mounted on paper with the correct species and location information on the label. If you feel like any of these steps are unclear, please consider re-watching this video.